The overall goal of this procedure is to produce clones in an embryo for the purpose of examining the autonomy of a gene or protein of interest. This is accomplished by labeling donor embryos with rhodamine dextran by microinjection at the one-cell stage. As shown in the companion to this video, microinjection of Medaka embryos. The second step of the procedure is to develop both donor and recipient embryos to the correct stage. The third step of the procedure is to dechorionate both the donor and recipient embryos. The final step of the procedure is to transplant cells from the labeled donor embryos to the recipient embryos. Ultimately, results can be obtained by analyzing the distribution, proliferation, and differentiation of clones of transplanted cells through detecting transplanted cells by their fluorescence or lineage tracers.
Hi, I'm Porazinski from the lab of Dr. Makoto Furutani-Seiki in the Department of Biology and Biochemistry at the University of Bath.
I'm Huijia Wang, also from the Furutani-Seiki lab.
Today we'll show you a procedure for cell transplantation in Medaka fish embryos.
We use this procedure in our lab to study whether a gene or mutation of interest has a cell or non-cell autonomous function. So let's get started.
When dechorionating Medaka eggs, use sterile solutions and tools to enhance the success of long-term observation and culture. Place a piece of P2000 grit waterproof sandpaper in the lid of a 9 cm non-adherent Petri dish. Use a wide-mouthed, fire-polished Pasteur pipette to transfer up to 10 freshly unclustered, cleaned and labelled eggs from a dish of egg medium to the sandpaper. Remove excess.